Hi, my name is Muhammad Adam Hakimi bin Kamal Zaman. My matrix number is E25A3989. Our group represents about microorganisms that cause food spoilage. Hi, my name is Muhammad Adam Hakimi bin Kamal Zaman. My matrix number is E25A3989. I will start the presentations with introduction of microorganisms. What is microorganisms? Microorganisms, or we call it microbes, are microscopic organisms, which means that they are too small to be seen with the naked eye. Also, microbes are smaller than human cells, and to see the microbes, we have to use microscope to see that. Microbes are the oldest life form on this planet. There is fossil evidence that living 3 billion years ago. That means microbes already lived on this planet in long time ago. Also, microbes can cause disease are called pathogens. While it might seem that they make up a large proportion of the microbes on our planet. Human pathogens accounts for less than 1% of microbial species, and microorganisms represented in the three domains of life, which is archaea, bacteria, and eukarya, which is living thing. Archaea have been found in the most extreme environment on Earth. Bacteria have found almost everywhere on these planets. And for eukarya, are multicellulars with diverse forms such as fungi and plant. Viruses are the only microorganisms that are non-living. Viruses can infect hosts like humans, plants, or animals. Microorganisms can be classified as prokaryotic or eukaryotic. For prokaryotic cells, lack of nucleus, and prokaryotics divide two groups, which is bacteria and archaea. And then for eukaryotic cells contains nucleus and other membrane-bound organelles may be either single cells or multi-cells. And microbiologists can answer some big questions which affect uh, many in our life. Why I say that? Because study microbes help us to understand our worlds and our place within it.
Good morning to Dr. Suganti. My name is Nurul Imam binti Muhammad Taufik. My number matrix is E25A3935. So I'm going to present my part, which is the introduction of yeast, fungi, and bacteria. The first one is yeast. Yeast is eukaryotic microorganisms belonging to fungi and typically reproduce asexually from budding or fusion. Usually, yeast can grow on the surface of the fruit skins that have begun to rot, and also can be grown in chemical-defined media such as solid agar. Yeast is commonly used in food manufacture such as fermentation and leavening agent. During fermentation, yeast was used in order to make alcohol such as beer and wine, since the fungi feed on sugars. Meanwhile, yeast can also be used as leavening agent for baking bread. The second one is fungi. Fungi or fungus are eukaryotic organisms and were included in the plant kingdom. For reproduction, fungi are unique because it produces both asexually and sexually. The asexual cycle produces metaspores, while the sexual cycles produce meiospores. The habitat of fungi is in soil that has a high organic content or dead things such as rotten wood. Fungi such as mushrooms or truffles are often used in food industry such as producing Epicurean delicacies. Fungi were also used in medical industry. A fungus called ergot was used in drugs that can induce labor in pregnant women. Last one is bacteria. Bacteria are commonly known as single-cell microorganisms. Why? Because bacteria lack of nuclear membrane and active in metabolically. Bacteria can reproduce by binary fission, which is an asexual process where a single cell will be divided into. Bacteria can live in extreme temperatures such as a hot and cool temperature than human, but the bacteria will grow better in room temperature. Bacteria are always used in our daily lives as we cannot live without bacteria, such as in medical or food industry. In the medical field, bacteria were used to make drugs such as antibiotics and vaccines. Meanwhile, in the food industry, bacteria were commonly used to ferment foods like cheese, yogurt, pickle vegetables such as kimchi.
Good morning. I am Muhammad Naizul Azim bin Abdul Hakim Amer, holding metric number E25A3901, and I will be presenting the environmental factors that accelerate food spoilage. There are four environmental factors that will accelerate food spoilage. The first factor is temperature. High temperature, um, 5 degrees Celsius to 65 degrees Celsius will encourage the growth of microorganisms. As a result, food will become moldy or rotten. Uh, Look at temperatures range of 5 degrees Celsius to 65 degrees Celsius. This is range is known as the temperatures danger zone, where the growth of microorganisms such as bacteria and fungi is maximized. When food is stored at high temperatures within this zone, these microorganisms will multiply rapidly. The result, our food will become moldy or spoiled in a short period of. Next, moving on the second factor, which is moisture or humidity. Microorganisms need water to live and reproduce. Therefore, a humid environment causes food to quickly become moldy, mushy, and slimy. Microorganisms, just like any other living things, need water to survive and multiply. Therefore, a humid environment causes food to quickly become moldy, soft, and slimy because the high water content accelerates microbial activity. Next, the third factor is oxygen. The presence of oxygen can promote the growth of microorganisms because the presence of oxygen can accelerate the oxidation process and the growth of fungi. This this oxidation process can damage the nutrients and fats in the food, making it quickly stale and unfit for consumptions. Right. Finally, we move to factor number 4, which is light. Food exposure to light, especially sunlight, can cause nutrients damage. As a result, the food change color and taste. This this happens because sensitive vitamins like riboflavin, vitamin B2, and vitamin C are easily destroyed by light. That's all from me. I will now hand over the presentation to the next group member.
Hey, morning everyone. My name is Muhammad Muzammil bin Mukhtar and my metric number is E25A3983. Today, I will be presenting on the topic of microorganism formation that causes food spoilage. To understand spoilage, we first look at the four steps of microbial formation and growth. The first one is inoculation. The process begins with inoculation. This is where microorganisms land on the food from the air, contaminated surface, or water. They need two primary resources to start, water or moisture, and the nutrients found in the food itself. The second one is favorable condition. They require favorable condition. This means they need the right temperature, often room temperature, and the right pH level to actually thrive and begin growing. Once conditions are favorable, we enter the multiplication phase. Bacteria and yeast reproduce very quickly through a process called binary fission, which is splitting into two. Molds, however, reproduce using spores that spread widely. Finally, in enzyme production, the microbes release enzymes that digest the food externally. This leads to decomposition, rotting, souring, and molding, which is the visible spoilage we see. Now, we will look at what spoilage actually looks like, depending on the type of microorganism. Bacteria. Bacteria is often invisible until the spoilage is severe. Common signs include sliminess on meat, a sour taste or smell in milk due to a lactic acid production, and gas production that leads to bloated packaging. Yeast. Yeast typically result in a fuzzy or wet appearance. They cause fermentation, such as the production of an alcohol smell in fruit juice. Humans also see a pink or slimy patches on surface. Molds. Molds are characterized by fuzzy, thread-like structure. The main body is called mycelium, which is a network of threads or hypha that grows into the food. The spores are the colored powder, black, green, or white on the surface, and these are the reproductive structure. This is why we clearly see different colored mold spots. Finally, let's address a common observation, the loss of mass or the case of shrinking bread. The main cause, the decrease in the bread size or mass, is primarily due to two things, the breakdown of its components and the loss of water. Action 1. Microbial consumption. First, microbial consumption, mold and bacteria actively consume the starch, sugar, and protein in the bread as their food source. This action breaks down the physical structure of the bread. Action 2. Water evaporation or drying out. As the bread gets older and its structural integrity is compromised, the moisture content rapidly evaporates into the air, especially if the bread is stored incorrectly. The result. The result is the combination of the microbes eating the material and the bread drying out, which makes the piece of bread physically smaller, lighter, and harder, or what we call stirred. In summary, food spoilage is a predictable process involving inoculation, growth, and the production of the enzyme that decompose food. Recognizing the forms of spoilage, whether slime, souring, or mold, and understanding the mechanism of mass loss, like in bread, help us better preserve our food. That's all from me. Thank you.
Good morning to Dr. Suganti. My name is Nur Hidayah binti Muhammad Ali. Metric number E25A3970. Case study that we found or used for our topic is food spoilage, microorganism and their prevention by Sima Rawat. The first microorganism that causes spoilage in food is yeast, such as Zygosaccharomyces, tolerate high sugar and high salt concentration. Example of the food is honey, dried fruit, jams, and soy sauce. Second is Saccharomyces, which is producing gasiness, turbidity, and off flavor associated with hydrogen, sulfide, and acetic acid. Example of the food is fruit flavored yogurt, wine, and other alcoholic beverage. Last is Decara or Brettanomyces, produce volatile phenolic compound responsible for off flavor. Example of the food is alcoholic beverage and some dairy product. The second microorganism that causes spoilage in food is fungi, such as mold. Penicillium present in soil and plant debris. Example of the food is fruit, vegetable, and cereal. Some can attack processing food such as jams and margarine. Second is Physoglymys. Because of the high heat resistance of its spore causes spoilage on pasteurized juice. Lastly is Aspergillus, which is grow faster and are more resistant to high temperature and low water activity. Example of the food is grain, dried beans, peanuts, and some spice.
Hi and very good morning. My name is Aunista Metization. My metric number is E25A3963. I would like to continue my friend's topic in case study, which is called bacteria. Bacteria that are causes food spoilage has divided to two, which is gram-positive and gram-negative. First of all, I would like to talk about gram-positive. First, we have Thermoana bacterium that grows on canned or served food kept at high temperatures. For example, like soup that's sold in vending machine. And the second one is the Bacillus and Geobacillus that causes a flat, sour spoilage of high or low pH canned food with little or no gas production. For example, like putrefaction canned food products, early brewing of cheese, and butyric acid production in canned vegetables and fruits. Second bacteria that causes food spoilage is gram-negative. First, we have Pseudomonas that include fluences, fragi, ludensis, and bird flava. For fluences and bird flava, it's only appear in surface of fruits and vegetables, while fluences, fragi, and ludensis are in animal dairy food like meat, fish, and milk. For the second one is Enterobacteria that includes Salmonella, E. coli, Shigella, and Yersinia. First, we have Awilla carotivora that causing a soft rot on vegetable, while biogenic aminos in beer that have obscene bacterium, bacon and other kill meats that have Proteus and Serratia, and coleslaw has Klebsiella. These are the proof and pages that we use for our case study.